What's happening, everyone? This is the lecture on the respiratory system, chapter 17, and this is the final lecture of this semester. So if you've made it thus far, you have survived this online course. Well done. All right, let's continue. Your respiratory system is divided into two regions, an upper region and a lower region. The upper respiratory tract is mostly responsible for warming and humidifying all the air that you breathe in. Also, the structures in the upper respiratory tract are responsible for your sense of smell and your sense of taste, as well as chewing and swallowing your food. Familiarize yourself with the structures that are located in the upper respiratory tract. The lower respiratory tract are basically everything else, everything that sits above your diaphragm and is within the thoracic cavity. Air travels down through your trachea, through all the bronchioles and branches to the lobes in your lungs to then undergo gas exchange. This is all the responsibility for the lower respiratory system. Familiarize yourself with the structures that are considered in the lower respiratory tract. Let's walk slowly through all of the structures that we presented on the earlier slide. And these structures are what the air that we breathe passes through. So starting with the nose and the nasal cavity. In peaceful breathing, air enters and leaves your respiratory system through your nose. Now just inside the nostrils are small hairs called cilia, and these cilia filter out dust and large foreign particles. The nasal cavity is separated from the mouth by the bony structure called your palate. You were to reach your finger into your mouth and press on the roof of your mouth, you would feel this palate. Also, a vertical plate of bone and cartilage, called the septum, separates the nasal cavity into two halves, into right and left. The cavity is lined with epithelium-rich goblet cells, which produce mucus. Now, projecting from the lateral walls of each cavity are three bones called concha. You might remember this from the bone chapter. The concha slow the air down to warm it and moisten it before it passes through the rest of the nasal cavity. At the same time, dust sticks to the mucus, which is then swallowed. The branches of the olfactory nerve, which is the nerve that's responsible for your sense of smell, penetrates the upper nasal cavity and goes straight to the brain. The sphenoid sinus, which you'll see is shown here, and you can remember from the bone chapter, and other paranasal sinuses, they drain mucus into the nasal cavity. Just behind the nasal and the oral cavities is a muscular tube called the pharynx. Commonly called the throat, the pharynx has three regions. First, you'll see the nasopharynx. Air only passes through the nasopharynx. This lies just behind the soft palate, which basically is behind the hard palate and the roof of your mouth. And the nasopharynx contains openings for the right and the left auditory tubes. So this is basically why you feel your ears pop when you go up in altitude or go down to sea level. The next region of the pharynx is called the oropharynx. Food and air passes through this region. So this is the space between the soft palate and the base of the tongue. It contains the palatine tonsils, which are the ones that are most commonly removed by a tonsillectomy, as well as the lingual tonsils that are found at the base of the tongue. And the last region of the pharynx is the laryngopharynx. This passes dorsal to the larynx and connects to the esophagus. Food and air also pass through the laryngopharynx. Now for the larynx. So the larynx is basically this small area between the hyoid bone and the top of the trachea where sound comes from. It's where your voice box is. And it also provides the division between swallowing and breathing. If you try this, try to swallow and breathe at the same time. You'll realize that you have to do one or the other. And the larynx is what controls this. The epiglottis is what allows this division between breathing and swallowing. The epiglottis is basically a piece of cartilage that covers the top of the trachea when you swallow your food. So this directs the food down to your esophagus rather than into your respiratory system. Also in your larynx, you have this really big piece of cartilage called the thyroid cartilage, and that's what is known as your Adam's apple. Down at the most inferior portion of the larynx, just before the trachea, is another opening called the glottis. Basically, two folds of cartilage line the glottis and allow it to close and allow it to open. The superior pair of folds is called the vestibular folds, and they work during swallowing to keep foods and liquids out of the airway. The inferior pair of folds are what your vocal cords are. They basically get smaller and narrower or wider and further apart to produce sound when air passes over them. This is how you sing and talk and have your voice. And that opening between those cords is called the glottis. So if you want to see something pretty cool but kind of gross, there is a YouTube video where Aerosmith, the musician, had a camera go down his throat while he was singing, and you can actually watch the vocal cords work. It's kind of graphic, but if you're really interested to see how all this works, I think it's a good video. So check it out on YouTube. Basically, you could just say Aerosmith vocal cords on YouTube and it should show up. Now let's talk about the structures of the lower respiratory tract. Starting with the trachea actually lies in front of or anterior of the esophagus, which is where your food goes. The trachea is a rigid tube, and it's about 4.5 inches long and about 1 inch wide. You'll notice on the slide it has these rings. They're cartilage rings that encircle the trachea to give it reinforcement and keep it from collapsing. The rings are actually C-shaped, and the open part of the C faces posteriorly towards the back, giving the esophagus room to expand during swallowing. At the base of the trachea, you have what's called the carina. At the carina, the trachea branches into two primary bronchi. The primary bronchi is shaded green in the right lung. So the primary bronchi are also supported by C-shaped rings of cartilage, and the right bronchus is actually slightly wider and more vertical than the left bronchus. And this makes this the most likely location for food that you've accidentally gone down your respiratory system or small particles to get lodged. So if you accidentally aspirated food and started choking, this is likely where it would end up. Immediately after entering the lungs, the primary bronchi branch into secondary bronchi, and this branch happens for each lung's lobe. You'll see 2 on the left and 3 on the right. These are shaded yellow in the right lung. The secondary bronchi branch again into smaller tertiary bronchi, which are shaded orange in the right lung. The cartilaginous rings surrounding the bronchi become irregular and eventually disappear in these smaller bronchioles. The tertiary bronchi continue to branch, resulting in very small airways called bronchioles, which are shaded purple in the right lung. The bronchioles divide further and further to form thin-walled passages called alveolar ducts, basically the terminus of these bronchioles. The alveolar ducts throughout the lungs terminate into clusters of alveoli. These are called alveolar sacs. And this is the primary structure where all of the air ends up and gas exchange happens with the capillaries. All of these lung passages that we talked about, they all exist to serve the alveoli, because it's within the alveoli that gas exchange actually occurs. So the alveoli are wrapped in a fine mesh of capillaries, basically a capillary bed, and the extremely thin walls of the alveoli and the closeness and thinness of these capillaries, they allow for efficient gas exchange. The exchange of air occurs through the respiratory membrane, which is basically the alveolar epithelium and the capillary endothelium. The inside of each alveolus is coated with a thin layer of fluid containing surfactant. Surfactant is kind of a soapy substance that breaks up the water tension that's on the inside of the alveoli. Water molecules are attracted to each other, and if left alone and not broken up, then they would draw the whole alveolar sac towards itself and make it collapse. So this surfactant creates a break to reduce that surface tension of the water, and this keeps the alveolus from collapsing as air moves in and out of during respiration. Interestingly, the COVID-19 virus is the way it would affect the lungs was it would destroy the cells that produce this surfactant. So this would cause dysfunction in the alveoli and poor gas exchange and then respiratory distress symptoms. Your lungs take up the space from just above the clavicle all the way down to the diaphragm and fill up the pleural cavity. The primary bronchi that come off the bottom of the trachea, as well as the pulmonary blood vessels that leave the heart, they enter each lung through a slit on the lung's medial surface. This slit is called the hilum. Your right lung is quite a bit shorter and broader and actually overall larger than the left. And it has 3 lobes to it, a superior, middle, and inferior. Because of its size, it handles 55% of the gas exchange. It's not equal between left and right. The right lung contains 2 fissures that divide these lobes. The horizontal fissure divides the superior and middle lobe, and the oblique fissure divides the middle and the inferior lobe. Your left lung only has 2 lobes, a superior and inferior, and only contains 1 fissure, the oblique fissure that divides those 2 lobes. So the reason your lungs are not symmetrical, and that one is bigger than the other, is to make room for your heart. If you remember, your heart sits a little left to center in that cavity called the mediastinum. And this takes up space a little left to center in the lung cavity. So this is why your left lung is a bit smaller than your right lung. The pleura are basically slimy, mucus-covered membranes that cover your lungs all the way around. And there's layers to it, just like everything else. The parietal pleura is the outermost layer, and this is actually attached to the inside of your rib cage. The visceral pleura is the innermost layer and lines all of the lobes surrounding the lungs. And in between the parietal and the visceral pleura is a cavity that's filled with fluid and creates potential space for expansion of the lungs and lubricates the surface. This cavity also creates a pressure gradient lower than atmospheric pressure. What is pulmonary ventilation? Well, basically, pulmonary ventilation is a fancy way of saying breathing. And breathing is the process of inspiration of air and expiration of air. Just like the transport of materials across tissue membranes, the action of inspiration and expiration happens as a result of a pressure gradient. Air moves from an area of high pressure to an area of low pressure. Also, there's quite a few skeletal muscles that support the action of breathing, and these muscles are called the respiratory muscles. Let's talk about these respiratory muscles. Starting with the biggie, the flat muscle that separates your thoracic and your abdominal cavities is your diaphragm. The diaphragm has a major role in expanding and compressing that thoracic cavity. When the diaphragm contracts, it actually pulls downward and flattens out. This happens during inspiration. Also during inspiration, the external intercostal muscles, the ones that are in between the ribs, they will pull the ribs upward and outward as they widen the thoracic cavity. The internal intercostals help out a little bit here as well. They elevate the ribs. So if you take a deep breath in and then let it out, you'll realize when you breathe in, your chest gets bigger. And these are the muscles that make all of that happen. They pull on the bones and expand that thoracic cavity. Now for expiration, instead of other muscles contracting and pulling everything back down, the diaphragm and the intercostal muscles just relax. And they go back to their original position, and the thoracic cavity reduces in size. Now this is all for normal, peaceful breathing, the breathing we're doing when we're at rest. But sometimes we breathe forcefully, such as in heavy exercise or labored breathing from a disease or an injury. What happens here is we have accessory muscles that kick in to assist these other respiratory muscles we just talked about. For forceful inhalation, the accessory muscles involved are the sternocleidomastoids, as well as other neck muscles, and your pectoralis minor. The neck muscles help with forceful inhalation by elevating the rib cage, and the pec minors expand the rib cage. Now for forceful exhalation, like if we were going to try to blow up a balloon, the rectus abdominis and the external obliques contract and they pull down on the ribs and the sternum, reducing the chest size. Try this out. See if you can feel all the muscles that move during forceful inhalation and forceful exhalation. All of these muscles for breathing are skeletal muscles, and therefore they require neural innervation to stimulate contraction. In our brains, specifically in the regions called the medulla and the pons, there are two interconnected breathing centers. You've got the inspiratory center and the expiratory center. So this is why we can breathe even when we're unconscious. These centers in the medulla and the pons keep controlling our breathing. The inspiratory center, which is in the medulla, is the primary controller and command center. This part of the brain sends an impulse to the intercostal muscles and the diaphragm to contract. Then these muscles contract and we inhale. The signal suddenly ceases, which allows these muscles to relax, and then in turn, we exhale. So this is what's happening all the time. So the inspiratory center controls inspiration, and indirectly it controls expiration. Now, in the pons, you have the apneustic and the pneumotaxic centers. These act on the inspiratory center to control the rhythm of your breathing. So all three of these centers work together during inhalation. And then the expiratory center, also in the medulla, this sends impulses to the abdominals and the accessory muscles when you're doing forceful exhalation. So say in the instance when you're blowing up a balloon, this is when your expiratory center will engage. Otherwise, when you're just normally breathing, the inspiratory center, the apneustic and pneumotaxic centers are all working. So how does the pons and the medulla in the brain know exactly what rate and what rhythm of breathing the body needs? Well, there are sensory receptors throughout your body that sense all these things up on the slide and then send signals to those respiratory centers on the state of what the body's situation. Let's start with the first influencer to breathing, oxygen. In your carotid and your aortic vessels, chemoreceptors exist that detect when the blood levels of oxygen are low. They signal this message to the medulla, and the medulla makes us increase our rate and depth of respirations. This brings more oxygen into the body and levels it out. Also, there are other chemoreceptors in the brainstem. These receptors detect decreasing pH levels, meaning more acidity, in the cerebral spinal fluid. Again, a signal is sent to these respiratory centers to increase the rate and the depth of breathing, to blow off any excess carbon dioxide, and therefore increase our pH back to normal. So let's think a little bit about what's happening when you exercise. Your muscles are extracting more oxygen because they need the fuel, and let's say glycolysis is in rapid flux to produce ATP for those muscle contractions. Hydrogen ions are one of the byproducts of glycolysis. So the drop in the blood oxygen, as well as the increase in hydrogen ions, are sensed by these chemoreceptors I just mentioned. And this causes our breathing rate to increase. This is why we start breathing harder when we exercise. So moving on, to prevent our lungs from stretching too far, basically to the point of damage, there are also receptors in the walls that will detect stretch. When they sense that stretch has gone far enough, they signal the respiratory centers to exhale. Also, when you are scared or you're surprised, your breathing responds, right? Your hypothalamus, as well as the limbic systems in your brain, they alter breathing in response to pain and emotion. And last, you have irritants. Nerve cells that are in your airways, they respond to things like dust or chemicals or super strong perfumes. This can cause you to cough or sneeze and then also influence your breathing. Let's go back to the topic of pressure gradients. Materials, air being one of them, they really like to move from areas of high pressure to areas of low pressure. The difference between the pressure in our lungs and the barometric pressure of our atmosphere is what drives our respiration. When the pressure in our lungs drops lower than it is in our atmosphere, air flows into the lungs, high to low, and we inhale. And vice versa for exhalation. At the top of our inhalation, the pressure in the lungs rise. And at this point, it will actually rise higher than the atmospheric pressure around us. And this causes air to flow out, and we exhale. Let's walk through this process of breathing step by step. First, our brain sends a signal to the intercostal muscles in the diaphragm to contract. They then enlarge the rib cage cavity, and attached to the inner part of our rib cage is that parietal pleura, the outermost layer surrounding the lungs. The visceral pleura, the inner layer, is attached to the lung tissue, and it clings to the parietal pleura because of that thin film of fluid in between. That's called the pleural cavity. The pleural cavity, since it's basically sealed off, it has its own negative pressure called intrapleural pressure. When the ribs expand and the parietal pleura pulls away, the intrapleural pressure becomes even lower, causing the visceral pleura to cling even tighter to that parietal pleura. It's almost like suction the way these pleural linings work. Now, this visceral pleura, which is attached to the lung tissue, it actually expands the lungs and enlarges all of the spaces within it. This causes the pressure in the bronchi and the alveoli, called the intrapulmonic pressure, to drop lower. All of this expansion causes a low pressure inside of the lungs, lower than atmospheric pressure. And then air from the atmosphere will rush in. And this is how we suck air into our lungs. Our muscles expand the space in the lungs, which make the inner pressure drop. Then the higher atmospheric pressure basically moves air into the lungs. If you can remember, our blood pressure works in the same way. It's also affected by the space within. The larger the diameter of the vessel, the lower the pressure. Same with the water hose. If it's constricted, that pressure is higher. If it's open and larger, this pressure is lower. This is how we expand our thoracic cavity to create the low pressure, lower than atmospheric pressure. Now we have filled all of the bronchi and the alveoli with tons of air molecules, which then creates increased pressure within. So now the high pressure is inside the lungs, and it's higher than the atmospheric pressure. So which way does air want to move now? It wants to move out. Normal expiration is a passive process where the diaphragm and the intercostal muscles just relax and the thoracic cage recoils back to its original size. Lungs are then compressed by the bones and air flows down the pressure gradient from the lungs out into the atmosphere. There are many factors that can affect the ease of airflow into and out of our lungs, but we're just going to talk about two of them here. First, pulmonary compliance. This refers to the elasticity of the lung tissue. Ventilation cannot occur unless the lungs and the thoracic cage can stretch and recoil. Diseases that cause scarring, such as tuberculosis or black lung disease, they make the lungs stiff, causing them to have difficulty expanding and therefore impairing ventilation. Next, alveolar surface tension. Again, the inner surface of each alveoli is covered with a thin film of water, which is necessary for oxygen and carbon dioxide to pass through the capillaries. However, water molecules are electrically attracted to each other, and if left alone, the water molecules would eventually pull together and collapse the alveoli. And obviously, if the alveoli collapse, gas exchange cannot occur. So to avoid this problem, alveolar cells secrete surfactant, which is a lipoprotein that disrupts the electrical attraction between the water molecules. This surfactant lowers the surface tension and therefore prevents alveolar collapse. Basically, any environmental toxin, such as smoke, pollution, chemicals, or particulates, they can have a negative effect on the production of this surfactant, as well as the compliance of the lung tissue. Doctors and clinical exercise physiologists can actually measure ventilation. They can measure someone's breathing to see if it's impaired or not. The device used is called a spirometer, which can hook up to a metabolic cart like a VO2 machine, and it measures inhalation and exhalation volumes as well as force. Here are some of the measurements that a spirometer can take. Tidal volume is the measurement of air inhaled and exhaled during quiet, peaceful breathing. This volume is measured in milliliters. I want you to take a deep, large breath in. Inspiratory reserve volume is the amount of air inhaled using maximal effort, just like you just did, minus the volume of normal inspiration. Now take a deep breath in and then forcefully push it out. Your expiratory reserve volume is all of that air that can be exhaled during maximum effort minus the normal expiration volume. So air will actually remain in your lungs after you exhale to ensure that gas exchange is still occurring. This amount is called the residual volume. It's kind of like that residual volume of blood that remains in the left ventricle after contraction. Same thing happens here. Normal residual volume is about 1,300 milliliters. Now, if you were to add up your tidal volume, your inspiratory reserve volume, and your expiratory reserve volume, you would have what's called your vital capacity. This is the total amount of air that can be inhaled and exhaled with the deepest possible breath. Now, if you were to add your residual volume to that vital capacity, you would have what's called your total lung capacity. This is the maximum amount of air that your lungs can contain. We have a picture now of how air actually gets into our lungs. Now let's go deeper and focus at the molecular level of how the oxygen and carbon dioxide gases actually exchange. Again, remember, these molecules always want to move from an area of high pressure to an area of low pressure. So how do you know what the pressure of, say, just oxygen is within the air? In order to know where the high pressure area is and the low pressure area is. To figure this out, you have to calculate what is called partial pressure. This is the contribution of a single gas in a mixture of gases to the total pressure. To calculate partial pressure, you multiply the percentage of that gas by the pressure. In the case of the atmosphere, it would be the barometric pressure. So the partial pressure of oxygen at sea level is 159.6, and you would notate this as PO2. And the partial pressure of oxygen in Gunnison is 120.5 PO2. So if you do the math that's on the slide, you'll see that the barometric pressure at sea level is 760 millimeters of mercury. And if you were to multiply that by 0.21, or 21% of the oxygen in the air, that's where you get that partial pressure of 159.6. Same thing goes for Gunnison. The atmospheric pressure in Gunnison, because we're at altitude, is much lower. If you multiply 574 by 0.21, you get that partial pressure of 120.5. So think about this. When someone first comes to Gunnison from sea level, their ventilation will increase, among other acute changes, but let's just think about ventilation. You breathe harder. Why might our bodies respond like this in relation to this partial pressure of oxygen at altitude? Basically, the lower the atmospheric pressure, the more reduction in the driving force of air down that pressure gradient. 159.6 is a greater driving force than 120.5, and therefore it's harder, our body will respond by increasing our ventilation. Now the partial pressures of oxygen and carbon dioxide, they vary from the air we breathe to when it's in our alveoli, to when it's in our arterial blood, to when it's in our venous blood. These variations in pressure are what allow the gases to move from one area to the next. So, for example, inside the alveoli, the partial pressure of oxygen is greater than it is in the receiving capillaries. And the partial pressure of carbon dioxide is greater in those capillaries than it is within the alveoli. This difference is what allows the gas molecules to then diffuse across the respiratory membrane. Turn your books to page 352, and I want you to walk through this process from the moment we breathe air in to where it goes to our tissues, and then it leaves, and it goes back out and leaves through our respiratory system. Pause the video here and take a look at that page. Basically, to optimally exchange oxygen and carbon dioxide, the pressure gradient must be adequate, the alveolar surface tension must be right, and the rate of ventilation must be in line. Any disruption to those factors will change gas exchange. So that oxygen has entered the blood now because of the pressure gradient being higher in the alveoli and lower in the capillaries. Now what? Well, if you think back to our blood chapter, what is the blood cell that is responsible for carrying the oxygen around? It's the red blood cell, the erythrocyte. And what structure within the red blood cell is responsible for bonding with that oxygen? It's the hemoglobin. So of all the oxygen that enters the body, about 1.5% gets dissolved in the blood plasma, the liquid part. And the remaining 98.5% binds to this hemoglobin, forming the oxyhemoglobin. 4 oxygen molecules per hemoglobin structure. Now this oxyhemoglobin travels to the cells and the tissues all over the body, ready to deliver its goods. Once it reaches its destination, that bond between the O2 and the hemi group needs to be broken before the oxygen can respond to the pressure gradient and move to the tissue. This is where pH comes into play. The pH of arterial blood is higher than the pH of venous blood. And this difference is what causes the oxygen to be released from the hemoglobin and therefore be free to pass across the cell membrane into the target tissue. So to summarize this, the oxyhemoglobin travels to the tissues, where the difference between the venous and the arterial pH breaks the bond, and the oxygen is released to travel down the pressure gradient into the tissues. The carbon dioxide in the tissue wants to leave. Here's what happens next. Because of the partial pressure of CO2 being higher in the tissue and lower in the veins, the CO2 passes across the membrane and enters the venous blood. 10% of that CO2 gets dissolved in the plasma, and 20% binds to those hemoglobins. Like oxyhemoglobin, there is a name for the hemoglobin that has CO2 molecules attached. This is called carbaminohemoglobin. The most carbon dioxide, 70% of it, is carried in the form of bicarbonate ions. This occurs when CO2 binds with water in the plasma, forms carbonic acid, then carbonic acid dissociates into bicarbonate and hydrogen ions. Most of this bicarbonate releases the CO2 at the alveoli, but some does travel to the kidneys, where it is filtered, and the final destination of the hydrogen ions is in our pee. This is what causes uric acid. So this concludes the lecture on the respiratory system, Chapter 17. Please email me if you have any questions at all, or we can FaceTime and go over any of these concepts. Thanks for listening.